I think I'll need to wear a jacket next time. God, it's freezing. Okay. Well, good morning, everybody. Can you hear me okay through this microphone? Or I've got a loud voice anyway, you can probably hear it anyway. Just make sure it's recording. So my name's Steve Matsas. I'm in the Department of Pharmacology, but I also work at the Institute for Respiratory Health. So my background is respiratory. And I'm going to start off today by giving you a talk on clinical pharmacology 1. So there'll be 2 lectures under clinical pharmacology. We're going to start today and we'll finish off next week. And if anyone needs to contact me at all in any way, you've got my email address that's written there. So firstly, what is clinical pharmacology? Well, effectively it's the study of drugs and biologics in humans. And the aim is to advance therapeutics in humans with mechanistic understanding of drugs and its actions, which is called pharmacodynamics, and drug disposition or pharmacokinetics. And pharmacokinetics is how the drugs enter and exit the body and how their concentrations change over time. So clinical pharmacology can be broken into 4 areas, which I'm going to discuss 3 of those. Drug development, that is clinical drug trials and how we test drugs in man. Drug utilization, or the therapeutics, and that's choosing the right dose and right drug for the patient. And clinical toxicology, identifying poisons in clinical cases and in forensics. And the last one is drug regulation. This is drug of choice. This is how they decide what drug is used within a country, in a hospital, which practitioner, which uses which drugs, etc. We're not going to talk about drug regulation at all. So we're only going to talk about the 3 main. But this is what clinical pharmacology covers altogether. So I'm going to start off by talking a bit about drug discovery and development. And effectively, you can think of when we start developing drugs, it starts in a pipeline where you look at thousands and thousands of particular molecules that might be important, and you have drugs for those molecules. So in this case, you've got, you know, this funnel where you start off with 10,000 compounds, and then it moves down to preclinical development, which is in animal models, etc. And you might take 250 of those compounds. And eventually you'll get really good ones, maybe out of that 10,005 that might go into clinical development. And from that, you'll then do your clinical trials, and we'll talk more about the clinical trials later on. And then you get maybe one of those drugs that will eventually get through to regulatory approval. Now, as you can imagine, there's a lot of work going to develop one drug from those original 10,000 compounds. And a study in 2020 estimated that the average cost of getting any drug from discovery to market is $1.8 billion. That's Australian dollars, so it's about 1.3 US dollars. And that was actually really interesting because the previous study had actually said it was about double that. And to be honest, you know, you look at those 10,000 compounds, what it's even got to get to that point is probably closer to, I reckon, $3.6 billion per drug. And you can imagine a drug company who's developed this particular drug has to get back all that money and make a profit. Hence the reason why some of these new drugs that you see are so expensive. You know, they can't just, you know, sell them cheaply, even though it might cost them very little. They're actually having to retrieve all that money that they've spent through all that development of that particular drug. And hence the reason, you know, like you have really high costs. So the drug discovery process is really important. And this comes from the point of actually starting where you have your target selection. You know, we're trying to understand the basic biology here. You then have a lead discovery where you might isolate a few things and actually get a few drugs that might be of interest. You have your medicinal chemistry, which develops those drugs to make them more therapeutically available. Bioavailability is really important to be able to get a drug to be absorbed, for example. You'll do in vitro studies where you'll actually examine what effect that particular drug will have on, on cells in culture. You eventually go into in vivo, which is animal models, and different animal models are designed for different type of diseases, and you'll test that drug in that system. And eventually you'll have this, you know, a few compounds which will eventually go into clinical trial that might eventually be your therapeutics. So clinical trials, the purpose of a clinical trial is to find a better and a safer way to prevent, screen for, diagnose or treat disease. And there are different types of clinical trials. So your treatment trials, which is, you know, the most obvious ones, is they test new treatments, new combinations of drugs, or new approaches to surgery or radiation therapy, or anything with people with a particular type of disease. So can they treat that disease? You then can get prevention trials. And prevention trials look at better ways to prevent the disease from occurring in patients. These are people who may have never had the disease or prevent it from actually coming back again. And these approaches may include, you know, medicines, vitamins, vaccines, minerals, or even lifestyle changes. You've got screening trials, and this is to test the best way to detect certain diseases or health conditions. And then quality of life trials, or maybe best supportive care trials. And these explore ways of improving the comfort and quality of life for those individuals with a chronic disease. So these are the sort of trials that you can do. And the thing is, there are both benefits and risks with having it being on a clinical trial. You can fully understand some people who may have a disease that there is no treatment for will go on a clinical trial because it's their only hope of actually getting treatment and maybe, if not save their life, at least extend their life. So the benefits then of a clinical trial is that you get new treatments, maybe better than those currently used, or maybe treatments that maybe a treatment that was never used, that's not available. Patients may be the first to benefit from these new drugs, which is great. The patients might also get high quality care. So you know that when you're on a trial, you're getting the utmost best care, because whoever's running that trial wants that drug to have the most opportunity to be able to be taken through to develop. And patients can help others. So by being on a clinical trial, the outcome of that trial, particularly if it's positive, will help others in future that will be able to get that drug. But of course, with these trials come risks. New treatments may not always be better than those that are currently used. New treatments may have really bad side effects, worse than the current treatment. Patients may have more doctor visits, procedures or tests to be done, which is going to take their time. And some costs may not be covered by trials, although, you know, in places like Australia, most of the costs are involved in the trial is completely covered by the trial. But there can be circumstances where it's not covered. So this must be taken into consideration when people go on to clinical trial. Not always will they be beneficial. In fact, a lot of clinical trials, most clinical trials, will be negative, particularly for new types of drugs, cancer treatments, etc. And there are a number of phases of a clinical trial. And we start off with our preclinical phase, which is down here. And this is, as I mentioned before, this is trying to get information on the action, the efficacy, toxicity, etc. of a particular drug. So this is all done in preclinical, in animal models, for example. Now, I've thrown one in here because this is not always used. This is a phase 0 trial. And a phase 0 trial is really to look at the pharmacokinetics, particularly like things like oral bioavailability of a drug or the half-life of a drug. And usually what they use is small sub-therapeutic doses in a very small number of patients, just to get an idea of whether this drug gets into the system and is at the right levels in these patients, and not so much whether they get an effect from the drug, but whether or not at least the drug is presented correctly. And this, though, is often skipped for a phase 1 trial. It all depends on the type of drug. But it's good because it can give preliminary data to allow a phase 1 trial. Then the phase 1 trial is usually involving between 20 to 80 people, and this is all about safety. These are given to healthy volunteers, so not people with diseases already. These people are just healthy people that basically you're trying the drug in them to see if there's any toxicity. Okay? So you want them to look to see if there's adverse effects. They also get information on things like the mechanisms of the drug, metabolism, excretion, etc. And these are often measured in phase 1 trials. But the outcome, the actual clinical outcome, is not as important. They usually look to see if they get any effect. But some of these drugs are done over a short period of time. They may not see effect, which requires much longer. It's all about safety. So phase 1 trials are safety. Then we go into the really important phase trial, which is the phase 2 trial. And now we're looking for efficacy. Does it actually work? Does it do something? So the goal of the phase 2 trial is to obtain this preliminary data on whether the drug works in patients who have a particular disease. And usually at this point it involves hundreds of patients. Again, they continue to look at safety, but most important is actually, does it work? Does it do something? And if it shows that it's doing something, then they'll go into a phase 3 trial. But to get to a phase 3 trial, it must be good. And the reason being is that now we're starting to involve hundreds or thousands of patients. And often these trials in a phase 3 are multi-center trials. In fact, you can have multi-center trials in phase 2. And multi-center trials mean is that it's in multiple places and often across the whole world. And one of the reasons why we often don't do it within, say, one country and we do it across the world is it depends on the disease type. So if you've got a disease which is not very common, you're not going to get enough people in one country at a particular time to be able to treat. So you have to do it in multiple places. Plus, if you're starting to look at hundreds or thousands of patients, 1 or 2 centers can't actually treat that many patients. So you have to spread it out. And the phase 3 trial is really to get more information about safety as well as efficacy. So it's to build on that knowledge. And only following a phase 3 trial can that drug then be reviewed, and it goes through the drug review application process where in this case it's American Slide, it's the FDA, the Food and Drug Authority. In Australia it's the TGA, that's the Therapeutic Goods Administration. They decide whether a drug is good enough to go through to the clinic. And they give permission and give authority for that drug to be used clinically. And there's a number of things that need to be taken into consideration. Is that drug going to be toxic? Is that drug going to be beneficial? And is it better than a drug that's already out in the community? Now, to be able to get approval, it doesn't have to be better than another drug, but at least it has to be as good as another particular drug out there. And then we have the last phase, which is a phase 4, which is called post-marketing. And this really can actually involve thousands and thousands of patients. So this is done over a long period of time, much, much later than the drug is actually being firstly put out to a clinic. Is to check the efficacy of that over a long period of time. And this is really important because sometimes, even though they've done drug trials over 2 or 3 years, it's only 10 years down the track that we see something bad happen with a drug. And so, there's been a number of drugs, particularly heart medications, which have shown to have negative effects down the track, in which case they then get removed from the system or modified, etc. So it's really important to have this post-marketing phase as well. So these are the main phases of any clinical trial. And it's really important to understand this if you eventually get into the clinical setting, or if you do research and more clinical research. The goal of anybody is to develop, in research, is to develop a new drug which is going to go into trial and eventually be used in the clinic. So what about how do we set up a design of a clinical trial? So first of all, we take a group of patients and they're assessed for their eligibility. As in, do they have the disease, for example, or do they have, if you're treating a particular drug for lung cancer, you may want them not to have had to smoked in the past. So you'll then decide what the criteria is for those patients to be eligible for the trial and what the criteria is for them not to be eligible. And then you exclude those that are no longer eligible. And then we come to a really important part. It's called patient randomization. If we just, selected your little friendly group of patients and treated them, that's very biased. You can't do that. You must randomize that population as best as possible so it's an unbiased cohort. And once you've randomized your patients, we'll talk a bit more about randomization in a minute, we allocate the patients to an intervention group or to a comparison group if you're comparing two drugs. And in each of these groups, you'll have placebos and you'll have drugs that you'll be analysing. And then you look at patient assessed then for your primary endpoints, whatever that may be. Now, randomization. There are two types of randomization: probability sampling and non-probability sampling. And first of all, we'll talk about probability sampling. So this is, your first one is your simple random sample. And the random sample is where you take the whole population, so everybody's accessible. And effectively, they're chosen in a way as like a lottery. A computer will select people based on nothing in particular. The second type of, and this is the best because it's completely random. However, we then go through a stratified sample. And this is where a whole population may be divided into subgroups, based on age, gender, diagnosis, etc. And then a random sample is taken from within this subgroup. And the reason for this is, for example, if you've got a minority of people, if you looked at the whole total population, you may miss that sort of minority. So you may want to include that minority in your study. So therefore, that's why you take these different groups and sample from within those groups as well. The third is the systematic sampling or interval sampling. And the subjects are chosen based on a systematic ruling. For example, you may have a clinic and you're doing clinical trial within your clinic. So you might say the last patient of every 5 patients will be treated on this trial. So this will be systematic, number 5, number 10, number 15, number 20, etc. And this is particularly good for an internal clinical trial within in a clinic. And then you get cluster sampling. And this is a multi-stage sampling. This is where population is divided on geographical location, for example, into clusters. And I'll give you an example. An example is if you want to look at school children. Now, you can't look at all school children, so you might put populations into, so you might say, right, well, 10 schools within Western Australia we'll look at. So these are populations based on the geography, and then you'll sample randomly within those particular schools. So that's called cluster sampling. And then you've got your non-probability sampling. And so the first one is convenience sampling. And to be honest, this is where often most clinical trials are run. And that is the subjects are chosen according to their availability and their accessibility. And it's, like I said, it's the most widely used method. You're going to use patients who are available to you. You're not going to just say, right, the general population's out there, therefore we can select anybody. It just generally doesn't tend to work that way, even though that is the best way. Again, it depends on the sort of study that you're doing. You've got purposive sampling or judgmental sampling, and this is selection based on particular characteristics. Now, the problem with this is that it could have bias to it because you're selecting certain people with certain characteristics only. So you're biasing your study. The third is your voluntary response sampling, and this is based on volunteers. So if you've got people go, yeah, I'll do it, you're biasing straight away to a particular type of person that might want to do that type of study. It depends, of course, on the type of study you're looking at. And then you've got snowball sampling or interval sampling. And this is where you say to somebody, well, do you know somebody who could do this trial? And they'll get somebody. And then you ask those people, can you get some more people? And it builds on from there. So these sort of non-probability sampling aren't the best, although let's be honest, convenience sampling is often used. So what about our actual study design? So there's two main design processes. One is a parallel trial, and that is you randomise your patients and then you put them into, say, a drug treatment group or a placebo group, and they'll run through that and they'll complete that trial and that'll be it. The other is called a crossover trial. Again, you randomise your patients and they'll go into, say, for an example, a placebo group and a trial group, a drug group. But then you'll have a washout period where they will not be on any drug for a period of time, depending on the half-life of the drug, etc. And they'll swap over. So the person that got the drug before now goes placebo, and the one that got the placebo will go on the drug. So the characteristics of the parallel study design is that the groups are assigned different treatments. They have usually a short duration of treatment. They're usually larger sample sizes because they're just running in one particular direction. There's no carryover effect because so you're not having someone having a drug and then it could influence the outcome when they go on placebo, as you would with the crossover. And it doesn't require stable disease or similar baseline. So that is, you're just taking them through from start to finish, end of story. The crossover design, though, is each group receives both treatments. So you need to have a longer duration. So if you're going to do both, the drug trial is going to go a lot longer. It's good because you don't have to have such a large sample size. So you can have shorter sample size because both groups of people are going to get the same treatments. They're going to get drug as well as they're going to get control. This is particularly good if you've got a very rare disease. So I work in the lung field, and we work on a disease called idiopathic pulmonary fibrosis. There are not a lot of patients with IPF. So therefore, having a crossover trial is probably more relevant to that particular drug, to that particular type of trial. You do have a carryover effect, which you have to make sure you minimise by having a washout. But you also require stable disease and a similar baseline because this disease or this treatment is going to go over a longer period of time. So we don't want the person to be cured halfway through because we're not going to be able to assess it. So we need that stable disease over a period of time. So now we're just going to go and talk a little bit about, I've talked about clinical trials, so we're now going to talk about therapeutics and where we go from there. So therapeutics really is trying to understand the right dose for the patient, the right drug and the right dose for each individual patient. And you can think of therapeutics as being a whole pathway where they get a particular drug and dose of that particular drug, and it's usually determined on population studies that have determined what's the most likely drug and dose that will work, but not necessarily, and we'll come into that later. It then goes through what we abbreviate as ADME. This is really important to know this. First of all, you have absorption of the drug, the distribution of the drug from the bloodstream into the tissues, metabolism of the drug, how it's broken down or how it's metabolized into active components or excreted components, and then excretion, it's eliminated from the system. And after that, you've then got a particular level of drug that's still in the system, in the blood, that can be measured. And that particular concentration will be in the blood and will be in the tissues, will be the concentration of drug that will actually interact with your target to give you either a therapeutic response, which is what we want, and in some cases, a toxicity, which we don't particularly want. And that will all depend on what concentration of drug is there and how a person responds to a particular drug. Now, we do know some things before we decide on the drug and the dose of drug. That is the knowledge before treatment. Things like the age of the patient, sex, size, the history of their response. So some people respond well to drugs, we know that. And they may even know that they respond well to this particular drug. So we can already judge and get some indication of what we should be using. We may know something about the genetics of the patient, their ability to metabolize a drug, the pharmacokinetics and the response, the likely response of that patient to a particular type of drug. We will know whether they are healthy or whether they have a disease. So, for example, if they have a disease in the target organ, or if there's a disease in one of the elimination organs, such as the liver or the kidney, then that drug can build up and it can get toxic effects. So, we have to be very careful if we know a patient's got disease, a lung disease, for example. Lung. Let me go back to lung again. If we've got kidney disease, as to whether or not they're going to be able to excrete it properly or whether it's going to build up in the body. And also other diseases, and other drugs and other therapy that may interact with each other. So if we know that particular drugs will interact in a negative way with each other or prevent them from being eliminated, then we can actually judge that when we decide the drug and dose. But as well as knowing things before treatment, we also have some knowledge during the treatment process. For example, the drug effect. If we give a particular drug and we can see that it has a clinical effect, we know it's working. An example of this, for example, a clinical observation, is if we're giving an antihypertensive drug for someone who has high blood pressure, all we do is we monitor the blood pressure, and after they've taken that drug, if the blood pressure comes down, we know it's working. So we don't necessarily have to actually we know that the drug dose is correct. However, if they get toxicities, then we know that that level may be too high. Likewise, maybe laboratory. Maybe we can't actually clinically identify the effect of the drug, but we can measure something in the laboratory to determine. And one of the things that we can, for example, is blood glucose. We can see that the blood glucose level is being reduced if they're on insulin therapy, so we know that that's working. The other thing we can do is we can actually measure the concentration of drug after it's gone through this ADME. And the importance of this is that this whole, the first stage of this pathway is very dependent on the individual. Everyone will have different absorption rates and distribution rates, etc. But if we can eliminate this by just going straight to the drug level when it's in an equal, stable state, so an equilibrium state, then we can get an idea of whether that level of drug is at the right level to have therapeutic benefit. And this is called therapeutic drug monitoring. And we're going to discuss this in some detail in a few minutes. So just taking this pathway in a little bit more detail, the first stage of this pathway, where you have your absorption, distribution, metabolism, and excretion, is called pharmacokinetics. And this graph on the right shows you what I'm talking about, where you have absorption and distribution. So you take a drug, that level of the drug increases in the blood until it reaches a peak. It's then distributed amongst the organs or amongst the tissues, and it drops as it's then slowly eliminated out of the body. Okay? So that's the normal distribution of a drug. Then the second part of this pathway is called pharmacodynamics. And this is the part where you actually get target interaction and therapeutic response. And you can see here there's a number of different graphs that can measure therapeutic response. This one is your Emax curve. And this shows you've got percentage effect up to maximal effect, and you've got concentration of drug, and it shows you that at a low level of drug you're not going to have much of an effect. As that concentration of drug increases, you get an effect until you reach an Emax. That is the concentration of the drug that you get maximum effect. And the important measurement amongst here, as well as known as Emax, is what we call your EC50. That is at the point, the concentration of drug where we get 50% of maximum effect on that drug. So this is your pharmacodynamics. And we're just going to go through a few definitions here which might help you understand, and it's probably good for your review as well. So pharmacokinetics then is the process of the uptake of drugs by the body, the biotransformation they undergo, the distribution of the drugs and the metabolites in the tissues, and the elimination of the drugs and the metabolites from the body over a period of time. In other words, what the body does to a particular drug. And note that we usually focus on the concentration of the blood in the blood plasma when we're talking about pharmacokinetics. Pharmacodynamics is the study of the pharmacological action on living systems, including the reactions with and binding to cell constituents, and the biochemical and physiological consequences of these actions. In other words, the effect that drugs have on the body. Absorption is the passage of a drug from the site of administration, whether that be through injection directly into the bloodstream, whether through the gut, from a tablet, into the plasma. The distribution is the process by which a drug leaves the bloodstream and enters the tissues or cells of the body. Metabolism is the process of transforming the drugs into different chemical forms by the body, either to be available to the body to then use and be effective, or to metabolize it for excretion. Excretion is the removal of the drugs from the body, and clearance, this is really important. Clearance is not the same as excretion. Excretion is removing the drug. But clearance is a pharmacological term which means the volume of plasma that all drug molecules need to be removed per unit of time to achieve complete elimination of the drug from the body. That is the clearance or clearance rate. Okay. And so know that there's a difference between excretion and clearance. Half-life of a drug is the time required for the body to eliminate 50% of that drug. Therefore, drugs with a short half-life are going to have to be administered frequently because they're not going to last very long. Drugs with a long half-life require less frequent dosing. And it's important to note that if there's a difficulty in drug excretion, if there's disease, etc., then there will be an increase in the half-life and the risk of toxicity. Therefore, it's good to know. And often, if you go for a test, say you're having a CT scan, and you have to have an enhancing drug where they're looking at injecting into you a particular marker which lights up and helps the CT scan to show the blood vessels and blood flow in the body, for example, they'll often ask you whether you've got kidney disease, because they need to know whether or not, if you've got kidney disease, whether that particular molecule they're injecting can build up to toxic levels. If you've got kidney disease, then they're going to be less likely to want to give it to you, or they're going to want to make sure that the levels that they give are not going to be toxic. The onset of action of a drug is the time between drug administration and the beginning of the therapeutic effect. The peak concentration of a drug is absorption rate, is where the absorption rate equals the elimination rate. Okay, absorption in, elimination out, that's the peak. And duration of action is the time for a drug to produce the therapeutic effect. Okay. So that's just a few definitions which I think are really important for you to understand when you talk about therapeutics. So now we're going to talk about therapeutic drug monitoring. We're going to talk about the rationale for therapeutic drug monitoring. That is, some drugs that need measuring and some drugs that you don't need to measure. When do you measure and what do you measure? And then dose adjustment, giving a loading dose and then a maintenance dose. Now, we're only going to talk about the first two in this lecture because it gets a little bit more complicated with dose adjustment, and that'll be talked about next week's lecture. So we're going to talk about rationale and also when and what to measure. So first thing to consider is the therapeutic range. And the therapeutic range is the concentration range where there is efficacy, that is, there is a response to the drug without toxicity, and that is without adverse events that occur in most patients. And this graph here shows you, you can see this is, shows you on this axis is the percentage of individuals that respond to a particular disease versus the drug dose. And there are three curves that we're interested in. One is the actual therapeutic effect. I'm sure this comes up. Yep. The therapeutic effect, then you've got the toxic effect, and in some drugs you may have a lethal effect. And these are really important because what we want to know is what point do most people respond to a drug but don't get toxic effects. And that area is called the therapeutic range. So some patients, for example, a small number of population, will actually respond well to a drug at really low concentrations. Most people require, say 50%, require a particular concentration, which is thought to be as a lower limit. And then some patients don't respond until you get really, really high concentrations of a drug. The problem is, once they have, you get up here with the high concentrations of the drug, then you're going to overlap with the toxic effects. And so the therapeutic range has to be the range where most people will be affected positively with a minimum amount of toxicity. I mean, here, for example, even at this particular range, at the 50% of the population responding well to a drug, there's going to be about 10% of the population that are going to have toxic effects. And of course, as I said, as you go higher up on that curve, more and more people are going to have toxic side effects. And at some point, there's going to be lethality with certain sorts of drugs. So the ideal dose then is within that therapeutic range, and this is defined as the target concentration. And this is the range that most people start when you get given a drug. Most people get given a drug in this concentration range. But this is great for population, but not necessarily for an individual. Because as I said, some individuals need higher concentrations of drug to respond, and some may not need as high at all. So population studies are good for starting a drug concentration, but we have to then look at the individual as determining where we go from there on. So what about the rationale for therapeutic drug monitoring? So what we're trying to look at then is we want to measure the concentration of a particular drug, particularly with drugs that have a narrow therapeutic range. That is, there's not a huge difference between, an efficacious drug where it's actually effective versus when it becomes toxic. If it's a huge range, then it's not so bad. But if it's really close, we need to make sure that that drug stays within that therapeutic range and not in that toxic range. And examples of this, for example, are of drugs that need monitoring. Digoxin, which is a cardiac arrhythmia drug and is also used for heart failure. Anticonvulsive drugs, immunosuppressants such as cyclosporin, sirolimus, tacrolimus, lithium, which is used for bipolar disorders, a number of different types of antibiotics. In this case I mentioned aminoglycosides, but there's a whole range of antibiotics that fall into this category, and different cancer chemotherapies such as methotrexate and busulfan. And this is because the toxicity, as you can imagine with some of these drugs, is really high, and therefore we must be very careful of getting the therapeutic dose and not in the toxic range. So we need to measure these particular types of drugs. But there are a number of drugs that we don't need to measure, and these are because the drug concentration adds little information. So for example, a drug that you can give lots of, and let's be honest, it's not going to cause many toxic side effects. I mean, paracetamol is one of those, I mean within limits of course, but people can take drugs and take them a little bit more often than they should, and they'll be fine. They're not going to have major toxic side effects. Other examples of drugs that don't require monitoring are those that you can measure clinically, so you can see an effect, so you don't need to measure it. For example, analgesia with morphine. If you give someone morphine, you'll know pretty soon as to whether it's affecting them or not. And because if they've still got pain, well, you need to increase the dose. But if they've not got pain anymore, you know that it's working well. As I mentioned, measuring blood pressure with antihypertensives, you can see whether or not that drug's working. Blood glucose with antibiotic drugs, antidiabetic drugs, and coagulation, looking at prothrombin time as a response to warfarin. We can measure those easy enough. We don't actually have to measure the drug concentrations in itself. And there are also a number of clinical indications for when we use therapeutic drug monitoring. So general indications are things, so drugs, for example, that have a high pharmacokinetic variability. So in some patients, we know some drugs can be very variable as to their levels in different patients. Therefore, if we don't know that it's going to be stable, we need to be able to measure those drugs. If we have a patient that's got renal or hepatic disease, there may be a problem with their drug elimination. And if we don't know the drugs are being eliminated, then they're going to build up potentially to toxic levels. So we have to check those. Nonlinear pharmacokinetics, I'm not going to talk much about that. We'll talk about that next week. Altered physiological states. Pregnancy is an example. If you're pregnant, you're going to respond very differently to drugs than when you're not pregnant. And likewise, you've got another life that you're looking after, and we do not want it to be toxic for the child. So we have to be very careful and monitor the drug levels to make sure that there's not going to be any sort of toxicity. And possible drug interactions affecting the levels. So, when you're taking multiple drugs, some drugs will affect the levels of other drugs, and we need to therefore measure and make sure that the drug is actually at the right level to be effective. And then there are specific clinical questions that may make us want to measure those drugs. For example, if we have treatment failure, the drug doesn't seem to be doing anything. And it could be due to patient non-compliance. In fact, that's the most common reason of treatment failure, is the patient doesn't take the drug. And so we need to measure it in the system just to make sure that the drug is there. It could be a failure due to absorption. So they're not absorbing the drug across, if they're taking the tablet across the gut, it's not getting into the system. And if that's the case, we need to measure it and make sure that they are getting the right amount of drug. And if a patient comes to you with another symptom, clinical symptom, we need to measure that drug concentration in case the symptom is not something irrelevant to that particular treatment, but it might be due to toxicity. So you want to make sure that it's not due to that drug being toxic. So again, you would measure the level of the drug in the system. So question then is, when and what do we actually measure? And how do we estimate the concentration of drug in the target organ, which is actually, or target tissue, which is actually going to have the therapeutic benefit? And to do this, we use an area under the concentration time curve, or area under the curve, which is AUC. So let's just first of all think about a one-off drug concentration. So in the red here, you can see that we've given a drug, that drug gets absorbed, it gets distributed, and it gets excreted. Okay, that's great, but we don't treat people like that. We don't give them usually one-off drug treatments. They have multiple doses and they do a course of drug. And when we give a course of drug, what happens is that drug's given, and we usually give the next dose of drug at around about that half-life of that drug, around the 50% mark, where the half of that drug is eliminated. We top it up with another lot of drug. And then when we do that, it gets increased again, and we get up to a bit higher level this time. And then we give it another dose, and another dose, until eventually we reach an equilibrium state where the maximum amount of drug is not going to change depending on how many doses we give that person. So the question then is, if we're going to try and measure a concentration of drug in the system to see whether or not it's at the right level to be efficacious, what do we measure? Do we measure at the peak of that drug concentration, particularly if you're worried about toxicity? So you might want to measure it up there. Do you measure it at the trough, at the bottom here, when you're trying to see, well, is the lowest level of that drug going to be still in the therapeutic range? Well, if we really want to know what the amount of drug is in that whole system, we really need to measure the total area under the curve to look at that particular drug concentration at time versus the concentration that we've been given it. So to measure this total area under the curve, we can't measure it using one time point. We have to use multiple time points, which, let's be honest, is not clinically practical. There are some cases and some drugs we do need to know this. We need to measure the area under the curve, total area under the concentration time curve. But in most cases we don't. So what time point do we measure? Well, we usually measure it at that point before the next dose. And I'll explain why. So single point blood sampling, what is the most informative time? As I said, we usually do it at the time before you take the next dose. And this is the reason why. So here on the left, we've got 3 different curves. This is plasma concentration versus time, and it depends on the route of administration. If we give a drug by intravenous injection, we're going to get a massive increase in the amount of drug in the system immediately, and that will then get distributed and peter off over time. If we give a drug by oral route or by intramuscular, there's going to be an absorption period of time where that drug has got to be absorbed, that then has to be distributed through the tissues and then eliminated. And the elimination, to be honest, as soon as you give a drug, some of it's going to be eliminated, so that really covers that whole time point. But the important time is we want to look at the point where absorption is completed and the distribution in the tissues are at equilibrium. And why is that? Well, we can think of drugs in a 2-compartment model. That 2-compartment model is that we give a dose of the drug and it goes into the central compartment. Now this is theoretical. It doesn't exactly happen exactly this way, but this is the model we use. And you can think that the central compartment is your blood system. Your blood, So if we measure the amount of the drug in your plasma, that will give us an idea of the drug in the central compartment. And that'll be slowly eliminated over time. But what also happens is that drug is then distributed into the tissues, okay? And that distribution goes into what we call the peripheral compartment. And the peripheral compartment, although it's not just the end product, or so the end tissue, it's really what goes out to the periphery. And so what we want, and it's that actually leads to the effect. So once it's hit your target, you then get your effect. But there's an equilibrium that's raised between the peripheral compartment and the central compartment. And that's the point that we really want to measure, because when we measure the central compartment, or the blood, that is reflective then of what the concentration of that drug is in that peripheral compartment. And that, when you look at this curve here, that's not down up here at the front of that particular path, front of that curve. It's down here where we're starting to get distribution and elimination. So it's down here that we need to measure. And we want to measure at the sample at equilibrium. That is typically before the next dose or the trough. So this is the graph I showed you before. This is each drug dose we're given at each half-life, mounting up till you get a steady state level. And this is the trough injection point here as you're about to give the next dose of drug. So when do we actually measure the drug? Do we give it after we've given one sample? Or do we measure it after we've given them two drugs, lots of drug? So if we think about some simple mathematics here, after we've given the first drug and we then give that at the first half-life, we've got approximately 50% of that drug in the system. We give the next lot of drug and then another half-life and the next lot of drug, another half-life, etc., until we reach a steady state. And so if we look at this, at the first injection, or first amount of drug, we've got 50% left at that trough. At the second dose of the drug, we've got about 75%. At third, about 87.5%. It's not until you've got about 4 to 5 lots of that doses of that drug do we reach that almost steady state level of close to 100%. And therefore, there's no point measuring drug after one drug sample or two. You measure it later on. In this case, around your drug, your fourth or fifth treatment dose. And there are exceptions to this, though. Exceptions are if you've got toxicity. If there's any question that that drug might be toxic, you must immediately measure the level of drug in the system. So it doesn't matter how many doses you're given, you measure it immediately. If there's a question about compliance, as you're not getting an effect of that drug, then you will measure that drug before you wait for steady state. And there's also specific times and specific situations that you might measure it. We won't go into that now. But there are other occasions that you might measure that as well. Okay, so what we've talked about today then is we've talked about that's good enough. You can measure it at that point. But, you know, you can measure it later on. In fact, you know, people might get measured every 6 months or something if they're on long-term drugs. But in theory, it shouldn't change once you've reached that plateau. And as long as you keep taking the drugs on time. And that's what happens if people don't take their drugs. Then you're going to get this up and down levels of drugs, and that's really not very good when you talk about therapeutics.
Okay, I see.
Mm-hmm.
Thank you.
So it's not a magical number. Remember, this is an example. And it might be that you have to go 6 or 7 times later. But it depends on the drug. But usually, by about the 4th or 5th dose, it's usually kinetics has reached equilibrium, and usually, you know, that's good enough. So often if you're taking a drug and they want to check you, you say you take a drug every day, well, at the end of the week, they might say, well, we'll now measure that drug, because effectively it should be steady state, it should be level. It shouldn't increase or decrease from that point on.
Okay. Thank you for clarifying.
That's all right. Okay.